I'm Knowlton from the University of California in Davis, and today I talked about mitochondrial fission and fusion in heart failure. These are recently described changes in mitochondria, which we used to think of as static energy factories, but have now found divide and fuse over and over again. In the yeast, this happens many times, even in an hour, and in humans and other mammalian cells and organisms, it likely takes much longer than this because of the more complex structure of these cells than in the yeast. However, we do know that fission and fusion are essential for normal mitochondrial function, which means they're essential for our survival. And originally, we looked at mitochondrial fission and fusion proteins in heart failure and found that in a rodent model of ischemic heart failure, that OPA1, which is optic atrophy 1, was significantly decreased. And looking at human heart failure, we found that human heart failure of the same type arising from having had coronary artery disease was also associated with a decrease in OPA1, but two genes involved in OPA1 helps in the fusion, and two genes involved in fusing the other part of the mitochondria, which has a double membrane, were increased. And in the non-ischemic human heart failure, we found an increase in the two proteins involved in the outer membrane fusion and no change in the OPA1 that fuses the inner membrane. So there were differences in human ischemic versus non-ischemic heart failure, but some significant similarities to our rodent model. But remember, our rodent model will have heart failure much less length of time than humans, who we treat arduously, trying to prevent them having to need transplant, which is the source of our heart samples when the heart is removed at time of transplant. So from this information that we had, we thought that these genes might also cause a cardiomyopathy in humans that maybe had not been noticed, because remember, there are many people with heart failure which we don't know the origin of. And these genes, when they're mutated, often are associated with inherited neuropathies that cause blindness. And so there are a number of people with diseases such as Charcot-Marie tooth disease that have a mutation in one of the genes involved in mitochondrial fusion. And so we looked at mutation of OPA1 using a mouse model that was given to us by Dr. Vertruba at the University of Cardiff in Wales. And although she had initially found in just simple biochemical studies, just like in fact, just weighing the heart, that the heart was normal, we found with careful study that although in the 3-month-old mouse, the heart was normal, and I should mention that if both copies of the gene are damaged, it is lethal in the embryo. So we're looking at a heterozygote form. And at 3 months, the heart was normal, but at 12 months, we started to see signs of functional decline with a decrease in developed pressure, which is very important, it's what keeps us moving, and also a loss of any reserve in response to a beta agonist. And this correlated with significant abnormalities on the molecular and cellular level, including loss of a DNA copy number. The mitochondria contain their own little DNA, and when they lose that, that's very bad. And decreased expression of antioxidant genes, and a number of other significant morphological abnormalities in terms of the distribution of mitochondria and destruction of mitochondria, or partial destruction. And mitochondrial dysfunction is measured by abnormal complex function, which is part of the electron transport chain, as well as decreased mitochondrial respiration. And in particular, we had impairment of complexes 2 and 4. So there may be, one of the important things not only is that the OPA1 is decreased in heart failure, and this could contribute to some of the abnormalities we see in ischemic heart failure, but also that people out there with these inherited neuropathies may have associated cardiomyopathies that are not being missed, and it may also account for some of the cardiomyopathies of unknown origin that we now see.